Welcome to today's lesson on bacterial cells. In today's lesson, we will explore what makes a bacterial cell a prokaryotic cell and examine the functions of the organelles it contains. By the end of this lesson, you will be able to identify and describe the key components of a bacterial cell and understand their essential functions. So far, you have learned that cells are the basic units of all forms of life. You have also learnt the basic structures of prokaryotic and eukaryotic cells and their key differences. These include the fact that a eukaryotic cell is larger and contains a nucleus, whereas a prokaryotic cell is smaller and stores its DNA in a circular loop. Building on this foundation, today's lesson will enhance our biological understanding of the characteristics and workings of bacterial cells. Before we look into the specifics of bacterial cells, it's essential to understand that bacterial cells are examples of prokaryotic cells. Prokaryotic cells are simpler and smaller than eukaryotic cells. In this lesson, we are going to look carefully at the organelles in a bacteria cell. Remember, an organelle is a name for the small organ-like structures that you can find inside of cells. These structures carry out functions that help cells to do their jobs. Now let's explore the various subcellular structures, or organelles, found in bacterial cells and their functions. Firstly, bacterial cells do not have a nucleus. Instead, their genetic material is a single circular loop of DNA that floats freely in the cytoplasm. This chromosomal DNA contains all the instructions needed for the cell's functions, including the genes necessary for building proteins and replicating new cells. DNA's main function is to code for proteins required for cell functions and constructing new cells. In addition to chromosomal DNA, many bacteria have small, circular pieces of DNA called plasmids. These plasmids code for additional genes and are not part of the main DNA loop. Instead, plasmids can replicate independently of the chromosomal DNA. Plasmids often carry extra genes that provide benefits, such as antibiotic resistance. It is important to note that not all bacterial cells contain plasmids. Next, let's learn about the cell membrane in a bacteria cell. The cell membrane surrounds the bacterial cell, maintaining its integrity. It is selectively permeable, meaning it controls which substances can enter and leave the cell. This control is crucial for maintaining the cell's internal environment. The ribosomes are the sites of protein synthesis in bacterial cells. These tiny structures read the genetic instructions carried by the DNA and build proteins accordingly. Proteins are vital for various cell functions, from structural components to enzymes that catalyze biochemical reactions. In addition, some bacterial cells have long, whip-like structures called flagella that extend from the cell membrane. Flagella enable bacteria to move towards nutrients or away from harmful substances, giving them a form of mobility. It is important to note that they are only present on some bacterial cells. Each bacterial cell also has a cell wall surrounding the cell membrane. The cell wall is a rigid layer that provides structural support and protection to the cell. The cell wall in bacteria is made of a substance called peptidoglycan, which is unique to prokaryotes. The remaining area inside the cell is called the cytoplasm. The cytoplasm is the jelly-like substance where all of the cellular activities occur. It contains the enzymes and nutrients needed for cellular processes. Let's take some time to look at a diagram of a bacterial cell, where we can identify all the organelles we just discussed. In the middle of the bacterial cell, we can see the chromosomal DNA appear as a tangled loop within the cytoplasm. The plasmids are the smaller loops scattered around the chromosomal DNA. We can also see the cell membrane that serves as the inner boundary of the cell, and the ribosomes as tiny dots dispersed throughout the cytoplasm, where protein synthesis occurs. The flagella are the long tails extending from the cell body, but we must remember that not all bacteria cells have flagella. Finally, we need to identify the cell wall, which is the outermost protective layer. It is crucial to be able to identify each of the components of a bacterial cell, as well as their specific functions. Now, let's work through some questions to consolidate our knowledge of bacterial cells and their organelles. Here's our first question. Look at the provided diagram of a bacterial cell. Label the following structures: plasmid, cell wall, cytoplasm, and ribosome. Pause here to have a go. We need to start by examining the diagram and identifying the different components within the bacterial cell. To locate the plasmids, we need to look for small circular DNA apart from the main bacterial chromosome. This smaller loop is the plasmid. The cell wall is the rigid structure surrounding the cell, providing support and shape. To label the cytoplasm, we need to identify the jelly-like substance filling the interior of the cell. Finally, we need to identify the tiny dot-like structures within the cytoplasm, which are the ribosomes where protein synthesis occurs. Using this approach, you can accurately label and identify the different organelles in a bacterial cell diagram. Now, let's look at our second question. Match the following functions to the correct bacterial organelles. Pause here to have a go. Now, let's see the answers. Firstly, recall that protein synthesis happens in the ribosomes. Secondly, enzyme and nutrient transport occurs within the cytoplasm, where various cellular processes and metabolic activities take place. The structural support is provided by the rigid cell wall through its shape and strength. Lastly, carrying genetic material is one of the functions of the DNA within the main bacterial chromosome. However, it can also sometimes be carried out in the plasmid, so we need to mention both. By understanding the roles of each organelle, you can effectively match their functions. Let's take a look at our third and final question. Provide two significant differences between a bacterial cell and a eukaryotic cell, such as a plant or animal cell. Pause here to have a go. Now, let's take a look at an example answer. One key difference is the presence of a nucleus. Remember that bacterial cells do not have a membrane-bound nucleus, with chromosomal DNA instead appearing as a tangled loop in the center of the cell. Comparatively, eukaryotic cells have a distinct membrane-bound nucleus. A second key difference is linked to the complexity and size of organelles. Bacterial cells have simpler structures without membrane-bound organelles, such as mitochondria or chloroplasts, whereas eukaryotic cells contain these complex organelles. Using these distinctions, you can clearly differentiate between the features of prokaryotic, bacterial, and eukaryotic cells. By walking through these questions and using diagrams to identify and understand the functions of bacterial cell organelles, you can tackle biology problems related to prokaryotic cells effectively. In today's lesson, we focused on understanding bacterial cells and their classification as prokaryotic cells, part of the wider topic of cell biology. We discussed the key characteristics that define prokaryotic cells, noting that they lack a nucleus and membrane-bound organelles, unlike eukaryotic cells. We examined the structure of a typical bacterial cell, identifying essential components such as the cell membrane, cell wall, cytoplasm, ribosomes, and genetic material, DNA, that is located in a single loop within the cytoplasm. Additionally, we talked about other features that some bacterial cells may have, including plasmids, small rings of DNA that can carry extra genes, such as those for antibiotic resistance, and flagella, which are tail-like structures that aid in mobility. We reviewed the functions of these bacterial cell parts, understanding that the cell membrane controls the movement of substances in and out of the cell. The cell wall provides structural support and shape. The cytoplasm is the site for various chemical processes, and that the circular DNA carries genetic information necessary for the cell's functions and replication. Through this lesson, we gained insights into the basic biology of bacteria, appreciating their simpler yet efficient structure. By grasping these concepts, you are better prepared to explore more complex biological systems and their interactions.
Watch the rest of the GCSE biology course at Sophos Education.